a series of very, very interesting diseases. And so misfolding can result in the aggregation, the clumping together, if you like, of otherwise soluble proteins. And some of these give these intractable amyloid deposits in cells and tissues. And we now know the number increases actually every year. There are at least 50 diseases associated with the amyloid phenomenon. The best known, and we've heard about, is Alzheimer's disease, but also the prion diseases, remarkably type 2 diabetes. It used to be called late onset diabetes. It now occurs regularly in children, even conditions associated with medical treatment, and Parkinson's disease, Huntington's disease, motor neurone disease, and so forth. And there's several interesting things about these diseases. The first is they're virtually unknown 100 years ago. Alzheimer's disease was first described in 1906 as a curiosity. The second thing is that each of them is associated with a particular protein or fragment of protein that forms these deposits. There are other proteins associated with them in many cases. And these can either be full-length proteins or fragments. They can be unstructured polypeptides or highly structured proteins. And so there's nothing in common between these that one can detect between the proteins that give rise to these diseases. So they're all amyloid deposits, and the best known disease, I say, is Alzheimer's disease. It's the most common form of adult dementia. Something like 40 million people suffer from Alzheimer's disease now. That number will increase to 130 million, it's predicted, in the next 30 years. And in fact, if you look at that whole set of misfolding diseases, you can estimate that in the not very distant future, perhaps 10% of the world's population will be suffering from one of these diseases. Type 2 diabetes, there are already 400 million people suffering from them, and they're incurable. And so this is a huge problem for the human race in the future. So in Alzheimer's disease, there's this A-beta peptide, that's one we've heard about before. It forms extracellular plaques in the brain. There's a protein called tau, which subsequent to this, it's thought, can aggregate within cells. And these give rise to toxicity that causes the death of neurons, the loss of memory, and cognitive skills that one is very familiar about with these symptoms. But this is one disease. This is Alzheimer's disease, but the symptoms can be extremely different. For example, if you look at this condition, if you put people on dialysis for kidney failure for 5 years, the dialysis is not completely effective. Not all proteins are filtered out. Their levels change in the blood, and every single person, effectively, who's been on this for 5 years will get deposits of amyloid, not in the brain, in this case in skeletal tissues and in joints, which cause enormous amounts of pain and discomfort. So these are amyloid deposits but in a completely different environment. There's another, just to give you an example, another type of disease is so-called cardiac amyloidosis, where amyloid deposits occur in the heart. And it's been recognized recently that perhaps 25% of elderly people who suffer from heart failure, it's exacerbated by the fact that these deposits restrict the movement of muscle tissue in the heart. There are other diseases. Here's a systemic disease where these deposits can occur in the liver, kidney, spleen, and so forth. Again, sometimes in huge quantities, kilogram quantities in this case, and these are fatal diseases. Normally the organs rupture from the load of aggregated protein. So the first question I want to say is, what are these deposits like when you get them in these diseases? Well, whichever disease you take, if you extract the deposits, whether it's from the brain or from the liver or from the joints or whatever, and look at them under electron microscope, the aggregates contain large amounts of these fibrillous structures. They're long, thin, they can be microns in length, typically 10 nanometers in diameter, lots of beta sheet structure, and they look essentially the same whichever disease you look at, even though the place of deposition, the symptoms are completely different. So why don't we want to get the structure? Well, first of all, it's clearly not functional. These are very ordered structures. They're actually weight for weight stronger than steel. You don't really want them in your heart. But even worse things happen, because we realized when we looked at these sort of early intermediates, these precursors, often called oligomers, you can see in the electron microscope, which form and then develop into fibrils. They're misfolded. They've got the wrong residues, if you like, for that protein on the sequence. They're very sticky. And we could show, for example, if you add them to various cell cultures, whether they're anything to do with disease or not, they'll penetrate cells and stick to various components or disrupt membranes. So amyloid formation doesn't just get rid of function, it doesn't just disrupt tissue and organs, it generates toxic species within the cells and the body. And so we put forward, this is very simplistic overview, this sort of unified view of all these diseases with all their different symptoms, and said under normal circumstances there are actually all sorts of what you can call housekeeping mechanisms. They scour our cells and pick up proteins which are misfolded or beginning to aggregate and target them for degradation. That's why we can continue in our functional states for decades. And it's the failure of these mechanisms that gives rise to pathology, either because you get loads of this stuff accumulating in your liver, for example, or because you get toxic species that can cause cell death, for example, in the brain. So this was our general view, and we thought, well, why are these diseases now occurring? And we realized if you look at the sort of quality control and the regulation mechanisms, these are all, if you like, modern diseases. Alzheimer's disease is occurring because we live a long time. The prion diseases, because particularly in Britain we were clever enough to feed young cows and old cows, which already had these aggregates present. If you don't eat and if you don't run around to catch your food and get lots of exercise, you can become obese, which is a modern phenomenon again, which is a huge risk factor for diabetes. And in the past, of course, we didn't put people on dialysis. It's a very good thing, but it ends up with forming these aggregates. So we thought these were linked to subtle changes in protein concentration, sometimes with mutations in familial diseases, or with long life where these regulatory processes fall, or with diet, for example. And this could then result in the aggregation of these proteins and ultimately a whole loss of the control system which ensures that proteins remain in solution. And I wanted to show a little movie which illustrates this very simply. What we did was to take Drosophila. I'll show you what this means in a minute. We put in there the 42 residue peptide, the human peptide, expressed it in the CNS. It doesn't do anything functionally in these Drosophila and the fruit flies. But we chose strains where you can see amyloid deposition and neuronal damage. And we then made mutations in the sequence that we predicted from our test tube experiments, if you like, would make this peptide a bit more aggregation prone or a bit less aggregation prone. For example, put a stickier group in or a charged group. And then this is a series of these. These are the wild type 4042. What you do is you put these in a test tube, the different strains, you bang the test tubes on the bench, the flies drop to the bottom. If they're happy, they climb up again. You get graduate students to count them as they climb up. And what's great about this is, first of all, this is increasing aggregation propensity. When you do the experiment, you can plot the graph straight away. You don't need to do anything else. You can see this amazing correlation. But it's a remarkable correlation because this, if you like, is physical properties. The aggregation propensity is a physical property. This is physics. It obeys the laws of physics. And this is biology. This is 20,000 genes in Drosophila trying to control this process. And the aggregation propensity changes by a few percent, but it's enough to cause the more aggregation-prone protein to cause massive neuronal damage. But if you make the peptide a bit less aggregation-prone, it's indistinguishable from not having it there at all. And you see this amazing correlation. Adding the small molecules, even at relatively low concentration, has a dramatic effect on the aggregation. If you choose the right small molecules, many have no effect. And you can get these huge effects by adding small drug-like molecules, in the test tube at least, that inhibit one or more of these steps, and different steps on the aggregation process. We generate a library of potential small molecules that we think are associated with A-beta. We select some of those, some are already drugs, some are new molecules. We look in experiments to see if these will inhibit the aggregation process. If they do, we look to see which step they inhibit. And so we get molecules which are potential inhibitors from this screening process. But this is in test tube. Does it work in living systems? Well, we are moving towards humans, but first we're looking at ones we can screen more easily. These are the C. elegans system. There's the nematode worm, which was mentioned actually in relation to plants. And we've actually set up an automatic system to follow the movements of these nematode worms. This is the C. elegans, which is transgenic for the A-beta 42. In other words, the A-beta 42 is being expressed in the muscle of the worm, and it causes paralysis. These are very, very unhappy worms. They're not doing much apart from sort of jiggling around. But if we add a small molecule, this actually is a molecule called bexarotene. It's actually already in use as an anti-cancer drug. And if we add that to the medium which the worms are developing, it has a really dramatic effect. And we have some very happy worms here that are moving around. This is all they do when they're happy. They just jiggle around like this. So it's fantastic news for demented worms. You know, we can treat them completely. The nice thing about this is it doesn't just apply to one of these amyloid diseases. We've looked, for example, at worms which express alpha-synuclein associated with Parkinson's disease. We found other molecules. This is a natural product called squalene. And we have many other molecules now, and if you put this in worms, they're extremely happy, and you've suppressed effectively the toxicity that generates from the aggregation of the alpha-synuclein. So it seems if this method works, it could be generally